Alright, Olia, let me jump right into this. This is June 2018, Biology, Unit 2, Paper 2. There we go. Question 1. Well, we have this thing everybody afraid here. Krebs cycle. Well, this whole oxidative phosphorylation thing. So we have an outline of the Krebs cycle is given in figure 1. That's what you're seeing here. With compounds labeled A to G and selected points in the cycle numbered 1 to 5. So they say, I already fill it out here, but you say write in your answer in figure 1. Insert the number of carbon atoms contained in each of the compounds labeled B to G. An example is given for compound A. Okay, so what I'm going to do is I'm going to show you a different diagram here. Then we'll come back to this. So if you look at this diagram here, so we can see that, well, the form that the sugar does enter into the Krebs cycle is called pyruvate. So this does actually come from the glycolysis and the link reaction. And you'll see it have a bunch of different things here. You don't need to memorize all the names, but it have 3 things that worth memorizing here. We have acetyl coenzyme A, citrate, and oxaloacetate, and just a bunch of immediates here. But what's notable here is that if we start, let me start with a 6 carbon compound called citrate, right? And what we see in here, we see in that carbon dioxide is being released at 2 points. So this 6 carbon thing turned into a 5 carbon thing. So there's a pentose sugar here. And this 5 carbon also, right, releasing carbon dioxide. So this is turning into 4 carbon. So it's 6, 5, 4. Right? And then you see it remains as at 4 for the rest of the cycle until it turns into something called oxaloacetate. And then the acetyl coenzyme A adds on 2 extra, right? The 2 extra carbons from the pyruvate. You see that one was lost to carbon dioxide. So this 4 carbon plus this 2 will become 6. So basically what you're gonna have here is, well, is a cycle of losing these carbons as carbon dioxide, right? And then regaining it at the end of the cycle from the pyruvate. So this 4 will turn into a 6. Now you'll see a bunch of other stuff here. This thing called NAD, and you'll see our next one called FAD. And you know, you might be like, well, what the hell is that? Like, you probably up to now you probably don't understand. Well, what NAD and FAD is, right? They're very similar. These basically, you could think of them as uncharged batteries, and what they do, they act like a glove to catch hydrogen, right, to become charged. And what can happen now is that when they charge, they actually have the ability to form ATP. That's just putting it very, very basically, right? And so when they catch a hydrogen, so this NAD becomes reduced NAD or NADH, and this FAD becomes reduced FAD or FADH2. And yeah, so these forms here, NADH and FADH2, these are like charged batteries. This is the uncharged one becoming the charged, and that is actually able to produce ATP during the Krebs cycle, but only one at a time. This is what you're seeing here. So this is called substrate level phosphorylation. Now that is basically what's happening in the cycle. You have a bunch of coenzymes named NAD and FAD and things like that, and all of those there are accepting hydrogens, ready to produce energy and ready to be, you know, that ready to carry on to the next step, which is called the electron transport chain, which we're not going to do here. But this is what the cycle looks like. So let me come back to the question. Right, so if we start from A, so this is the 2 carbons from the pyruvate, and here this is B, this is the citrate, so this is the 6 carbon, losing a carbon dioxide, so become at C 5 carbon, losing the next carbon dioxide, it becomes 4 carbon, and then it remains at 4 at D, E, F, and G. G would be oxaloacetate. And that's it there. and then it adds on the 2 carbons from the pyruvate again and it restarts. It's not a bad Krebs cycle question, not too difficult. Alright, so the next thing here is writing your answer in figure 1. Insert the input and product at the point numbered 4 as completed for the points numbered 1, 2, 3, 5. The points 1, 2, 3, 5 is these NADs, right? And I say these accept a hydrogen. Well, this one here, this is the only case of FAD. And what that does is that this actually accepts 2 hydrogens. So this becomes FAD H2. Alright, so the next question here is explain the 2 main roles of the Krebs cycle. Well, the main thing in this is to produce energy, right? At least for it to pass on to the next step. So one of the main things here is that it produces NADH and FADH2. Remember, these are like the charged batteries. NAD and FAD would be the NAD and the FAD. Those would be like uncharged batteries until they start accepting the hydrogens. Now what this does is that it facilitates production of ATP, which is of course the energy currency used by organisms for any metabolic processes. And here I just have like an explanation of how this happens. Feel free to read it, but one thing that's notable here is that this is a cycle. So the Krebs cycle of course enables the restarting of that cycle. So when it becomes this 4 carbon compound oxaloacetate, what happens there is that, remember, it will add on the 2 carbon compound from before, from the pyruvate to become citrate again. So it uses its products to restart the cycle. The last thing that we have here is that remember, ATP is also produced during the Krebs cycle itself. It doesn't have to wait until it reaches the electron transport chain. So 1 ATP is produced per cycle due to substrate level phosphorylation. That there is from the reduced NAD and FAD. So that's 2 main roles of the Krebs cycle. The rate of photosynthesis is measured in individual leaves of a plant species at different light levels, and the data obtained are recorded in table 1. Here you can see the data. They have the light level or the light intensity, and these are the standard units for it. And here we have the rate of photosynthesis in standard units. Now if you just look at the numbers here, of course we're seeing that the light level is steadily increasing and the rate of photosynthesis is increasing at first, but then you'll notice that it becomes more or less constant by the end, right? That there's a kind of saturation point. So they say on the grid provided in page 7, plot a line graph based on the data provided in table 1. And they say using the concept of limiting factors suggests 3 reasons for the shape of the graph drawn in B1. So the first thing we're going to do is we're going to look at that graph. Alright, so here we have the graph, and let me try to explain what's happening. Here you'll see the photosynthetic rate. Remember, this is the responding variable, and this here is responding to the light level. Now you'll notice this is a line graph first of all, and well, your marks for this is just plotting the points, making sure that they occupy enough space on each axis. There's a line graph, things like that. And that you label your axes. Remember, your responding variable goes on the y-axis and your manipulated one goes on the x. So let me explain what's happening here. Now you'll notice that right in the beginning here, we'll see that the rate of photosynthesis is increasing quite steadily and steeply as light level is increasing. So that means that at this level here, light is actually the limiting factor. So, which means that light, if there's no light, it actually limits the rate of photosynthesis. Now at a point here now, the rate is still increasing, but not at the same rate. Right? So what's happening here is that light is still a limiting factor, but there may be other limiting factors that keep it from increasing, at a faster rate. So, for example, there are other limiting factors would be like carbon dioxide levels, temperature, water levels, right? All those are needed for photosynthesis. Now, at a point now, we'll see here that, especially after about 1100 standard light units, we'll see that the graph sort of tapers or plateaus. So at this point here, light is no longer the limiting factor, because at this point here, if we add more units of light, it is not increasing the rate of photosynthesis. So the light is no longer limiting the rate of photosynthesis. What might be limiting it at this point might be actually the number of chloroplasts, or the amount of chlorophyll. There's just not enough chlorophyll to be stimulated by the light anymore. It could be other things too, like temperature and carbon dioxide, but at a point, light is no longer the limiting factor. And that's basically what this graph is. So let me look at the next question there. So they say using the concept of limiting factors suggests 3 reasons for the shape. So it's basically what I said before. Well, limiting factor is one that influences the rate of a reaction in such a way that it may hinder the reaction or prevent the rate from increasing. So as I said in the beginning, if you didn't have light, that would have been the limiting factor, right? That would have been preventing or limiting the rate of photosynthesis. In the graph, light is the limiting factor. At low light levels, photosynthetic rate is high and rapidly increasing as light intensity increases. So this is basically adding more light units, the rate of photosynthesis becomes higher. That makes sense. So that happens very early on in the graph, as you see here. But as we go on now, especially around this part where it starts to curve, light is no longer really being the limiting factor. What's being the limiting factor here is probably the number of chloroplasts. And towards the end of the graph as well, maybe at this point here, all the chloroplasts are being stimulated. You can't stimulate anymore, right? So you can't really increase the rate of photosynthesis anymore past that point. So at high light levels, the rate of reaction becomes constant due to light no longer being the limiting factor. And other factors such as temperature may also become limiting factors as the rate of reaction is increasing. So I'll say examples like if you have very high temperatures, well, you need enzymes to carry out photosynthesis, like Rubisco, for example. That's an enzyme. And if that begins to denature and the protein actually begins to warp and lose its structure, it also loses its function. So that would cause the rate to plateau or even drop. Alright, so figure 2a shows a cross section of phloem tissue, and 2b shows a longitudinal section of phloem tissue. The way to draw and label a diagram of the 2 phloem cell types shown in figure 2b. So the 2 cell types, and I'll put up a better diagram of it just now, but the 2 cell types that basically is seen here is, this is a sieve tube, right, or a phloem tube, or a sieve tube element, and next to it, which is what you're most likely seeing here or here, which you can see the nucleus. These are called companion cells, and what these do is that these supply the sieve tube with ATP, because these companion cells are actually filled with mitochondria. So they say describe 2 features of each phloem cell type shown in figure 2b. So the sieve element, it doesn't have a nucleus, it doesn't have mitochondria, it doesn't have any large organelles really. And end to end, what they'll have is these sieve plates, kind of like a sift, right? Kind of like a barrier with small holes to allow passage of sucrose. They have an elongated shape. Again, they got like tubes, and they have a large empty lumen to allow transport of sucrose or any assimilates that have to pass through. Kind of like how the xylem is hollow. Companion cells, well, these have nuclei, prominent nuclei, and they have many mitochondria to provide ATP for the metabolic needs of the sieve tube elements. And they usually attach to the side of the sieve tube, and their cytoplasm is quite dense. state the major function of each of the two cell types, right? So the sieve element is like a tube, right? So it transports sucrose from a source to a sink. A source is a photosynthetic organ like a leaf. A sink is a non-photosynthetic organ like a root. So it carrying basically to what have to what don't have, or what could make to what cannot make. It also transports like other assimilates like an amino acids, and it does this in a bidirectional method. Bidirectional means it could transport it up the stem or down the stem, unlike the xylem, which only could go up the stem, because the xylem relies on a transpirational pull. So the companion cell, what this does is that it facilitates the movement of sucrose into the phloem tube cells by diffusion. So basically you could think like loading the sucrose in and out, providing ATP to the subtube element to allow active transport and uptake of sucrose. So the companion cell is basically like the engine of the whole, the engines of the whole phloem. Think of it like that. Without the companion cells, you wouldn't have any translocation or any movement of these assimilates occurring. Alright, so we're still on question 2. So this is B. John is a 62-year-old male who recently began using the antidepressant fluoxetine to deal with the loss of his wife. His blood pressure and pulse rate are monitored for 1 hour after he ingests the antidepressant. 10 minutes into the monitoring process, John falls asleep. Blood tests show that 30 minutes after ingestion, fluoxetine enters John's bloodstream. And this figure here shows the data on 3 things. And the data is going to be his systolic pressure, his diastolic pressure, and his pulse rate. The pulse rate is like the number of, you think of it like the number of beats per minute for the heart. And so it are two times to note here, right? The 10-minute time, and this is where he fall asleep. I'll just put a little S by that. And the 30-minute period, this is where it enters bloodstream, and I'm just gonna put a little B by that. The fluoxetine enter the bloodstream here. So let me just observe what's happening here. Now, something I should note here is that John most likely suffers from hypertension, because the average, I don't know what it is as I get older, but as your average blood pressure is supposed to be 120/80, and we could see here that, well, his resting blood pressure, we could say, is 140/90 something. Right, this is before he take the antidepressant and things. So yeah, so he already have kind of elevated blood pressure. The pulse rate is actually pretty normal. Usually it's be, I think, from 70 to 100, and you know, it's 80. We can see here the fluoxetine is actually making it climb. So we'll notice here, we look at what happens when he falls asleep. So when he falls asleep, you'll see that his systolic pressure actually goes down from 140 to about 125 or so, a little more than that. So it actually has a dip. His diastolic pressure decreases as well, not as much, but it still decreases. And when he falls asleep here, his heart rate actually slows down. His pulse, slows down as he begins to fall asleep, and it sort of stays low here. But what's happening here is, as the fluoxetine, the antidepressant, is entering the blood, we'll see that there's a spike in his systolic pressure. There's also a general increase and sort of a spike. I guess we could say it's a kind of spike, or it's more like a hill in his diastolic pressure, but there's a definite increase. And not a spike in the pulse rate, but there's actually a steady gradual increase in the pulse rate until it reaches 120. And you know, that can be good for someone his age, for it to be up to 120 beats per minute. So let's look at the questions. So they say explain what is meant by the following terms, and the terms are blood pressure and pulse. So blood pressure, remember, is divided into systolic and diastolic, but the general definition for it is the force of blood pushing against the walls of the blood vessels due to pumping, usually against the arteries. So when this blood, remember, pressure is force over area. So the higher, the more force that the heart has to, the more strain that the heart has and the more force it has, it pumps out that blood. That is what is the blood pressure. Systolic and diastolic. So systolic, I used to remember it like S for squeeze. You could think of it like, imagine you're squeezing the bottom of the heart and the ventricles, and it's squeezing the blood out, right, out into the aorta and things like that. So the systolic pressure is the ventricular contractions during heartbeat. So this is the higher bits of pressure here. Diastolic is more like when the heart relaxes, when the ventricles relax and allows blood to refill the heart. So this is usually like a lower pressure. It's 2 marks, so just a general definition, and you could also put a small definition for systolic and diastolic, or at least compare them. Alright, so pulse. So pulse is I mean, everybody know what pulse is, but to define it is the kind of tricky thing. But the pulse is the throb that is felt in arteries due to the force of the heart pumping blood. So usually like when you feel your wrist or your neck and you feel in that throb, that is what the pulse is. So it's like the feeling of the surge of the blood in the arteries. And the thing is, you would only, to say I just put arteries, you would really only feel a pulse in the arteries, because that is where the blood pressure is high enough, where you can actually feel a pulse. The average pulse range is 60 to 100. So all of these here we just talk about, so I'll just read the answers. This is describe the effect of each of the following parameters on blood pressure and pulse rate. Well, if you go back to where I was talking about the graph with sleep, we said that systolic pressure decreases while diastolic pressure decreases minutely, so not as much. And the pulse rate decreases at first, but it may show slight increase or fluctuations after a few minutes. That's easy, just 1 mark. The fluoxetine causes all 3 parameters to increase, with a steep increase in systolic pressure and a gradual increase in pulse. Should John continue to take fluoxetine? Give a reason. Well, I think the general answer here is no. Again, it's because of the spike in blood pressure, especially the systolic one. And it's also due to his age. John is now a young man, and he already have hypertension. So if the pressure remains high, you have a high chance again of stroke. So the fluoxetine may result in his heart being overworked due to the increased pulse rate and the increased force against his arterial walls. Due to the increase in blood pressure may result in CV's cardiovascular diseases. Right, his blood pressure is already naturally high, so it might actually put him at risk for a stroke. I guess if you say yes, eventually if the antidepressants work, his overall pulse rate and his overall blood pressure might actually decrease and might be better for him in the long run. So we don't know, but so we only have limited information to go from. So but if you put yes, you would say that. Alright, so here we have the next question. Alcohol consumption can have short-term and long-term effects on the liver. In the box provided below, draw a flowchart to illustrate the stages in the detoxification of alcohol in the liver, including the enzymes and coenzyme involved. Alright, so I wouldn't really go into explaining it too much. What I'll do is that I'll just post up the flowchart. And what you want to do is you want to make sure that you have the general steps and at least one enzyme named. So the next question here is explain how the metabolism of alcohol can lead to a short-term effect on the liver. Right, so just to sum this one up again, usually NAD is used to oxidize fatty acids in the liver, to break that down so it doesn't get stored in the cells. Instead now, when you drink ethanol or alcohol, you drink your rum, it is being used remember, NAD is like a resource. You only have so much uncharged batteries. So the NAD is now being used to oxidize ethanol instead of fatty acids. And thus, fatty acids cannot be oxidized, can't be broken down quickly enough. So they begin to accumulate, end up getting stored in the liver cells. That results in inflammation and then scarring. Describe how alcohol-induced cirrhosis occurs and its effects on the liver. This is like scarring of the liver, right? Now remember, this is a non-reversible process. You really only could slow it down past a certain point. But how this happens is that the hepatocytes, as the liver cells, they die during damage or inflammation, and this again could be due to the accumulation of fats, and they get replaced by scar tissue. Just like again a cut on your skin, it's like again cuts in your liver, not literal cuts, but due to the death of cells. And this occurs due to long term. So one thing here is that it's long term excessive alcohol consumption. As a result, the liver function is severely altered. It can't secrete bile salts, it can't help detoxify the body, things like that. They complete table 2 by writing the numbers 2, 3, 4, 5, and 6 to indicate the order of steps in the replication of HIV in host cells. Steps 1 and 7 have been done for you. Okay, now I'm going to show you, I'm going to put a diagram, and the diagram here will help explain my answers that you see here. So let me see what's going on. First, imagine you have a mature HIV, right? So you have mature virus in the body, in the bloodstream. First thing it does, it infects a cell. How does it infect it? It basically penetrates the cell membrane. Then you have a process called reverse transcription. Think of reverse transcription as the rewriting the code of the cell. So it's kind of like it making multiple DNA copies of itself, right? Then it enters through the nucleus. So after it sort of rewrites its own code, it enters in its nucleus, then it integrates into the DNA of the cell. Right? So this is now putting that instruction into the cell so that it could build new copies of itself. Transcription is going to happen, and transcription is the formation of messenger RNA, and that there now is kind of like writing the blueprint to build new HIV proteins. Assembly is basically translation. So remember, transcription is followed by translation. Translation is where the ribosomes read the blueprint, they read the mRNA, and they build the protein as a result. So this is actually building new viruses. This is why it's called assembly. The new viruses are made here, and it's going to move towards the cell membrane, where it's kind of like exocytosis. It sort of buds off, and then it matures, and then it infects more cells. So that there is the process, and from what we see in here, the steps is going to be transmission, the actual placement of the HIV into the bloodstream, infection where it goes into a cell, reverse transcription where it's going to make multiple DNA copies of itself, integration where that is actually going to go into the nucleus and the DNA of the host cell, transcription where it's going to make the blueprint to build new proteins, HIV proteins, translation, the actual act of building the proteins by the ribosomes, and then the release, the maturation and release of these new HIV cells, HIV pathogens that could then go on to infect other cells. Alright, and here we have a relatively easy one. Describe 3 ways by which HIV can be transmitted. There's 3 marks, so 1 mark for each. First one is through, sexual intercourse, exchange of sexual fluids. As for example, semen and vaginal fluids, all of these here can transmit HIV. Blood transfusions from an HIV infected person to an uninfected person. And this could also occur during organ transplants. When when a baby ingests an HIV-infected mother's breast milk or colostrum, that could also transmit HIV from mother to child. It also could be transmitted from mother to child via the placenta during childbirth. And well, this fits under blood transfusions sort of as well, but like if you share hypodermic needles, like taking intravenous drugs or drug abuse, if someone who has HIV uses needle and someone who doesn't, little remnants of blood and the virus could of course transfer. Easy, any three of these would work. Alright, so let me look at question 4. For a top carnivore to be numerous in an ecosystem, it must feed at many different levels of the food chain and not just the one beneath it. Discuss the veracity of this statement. Veracity in this case, you could think of it meaning truth, right? How true it is. Well, it is true, because first we need to remember what's a trophic level. A trophic level, when you have a food chain, and let me say you have a food chain that is lettuce, rabbit, hawk. The hawk is at the top of the food chain, and that would have the least energy available, and the lettuce would have the most because it has the producer. So each trophic level, which is like a feeding level, has different amounts of available energy. The top carnivores would have the least energy available from the level beneath them, as seen, how a pyramid of energy is, where it's kind of like this, and the producers would be down here, and this would be your primary consumers, your secondary consumers, your tertiary consumers, and so on, if you have quaternary too. And you have less and less energy being made available. And this is because only 10% of the energy is transferred from one trophic level to the next. Because each time energy is being transferred, you get about approximately 90% being lost to the atmosphere. This occurs due to heat loss during respiration and excretion. So the 10% actually passes on through each trophic. So, for example, if you had 1,000 units here, I'm just making up a number, if you have 1,000 units available here, you would only have 100 units being available here per organism, and then you would only have 10 here, and so on. So if the organism at the top only feeds on this one that only has 10, it's not going to have enough. So typically it will have to feed at lower levels as well if it wants to survive. So the good thing is at lower levels you actually have a greater number of organisms. But these here down here would be the prey, and this is because there's just more energy available to them. There's more organisms for them to eat. There's usually these primary consumers are herbivores, and there's usually a lot of plants to eat, a lot of producers. Now there's a fewer number apex predators or top carnivores, because there's just less energy being they require a lot more energy than the ones down here. So what has to happen is that a predator's diet must thus consist of food from various trophic levels, not just the one below. And predators must expend a lot of energy to actually stalk and hunt their prey, right? And if they don't get that amount of energy to them, they will die out, they will go extinct, or they will have to migrate. So yeah, it's true, they have to feed from multiple levels. Okay, so here we have a kind of long question. I think this is 9 marks, right? So we're going to go through it. Within any ecosystem, biodiversity exists at 3 levels. We have the genetic diversity, species diversity, and ecosystem diversity. Discuss the interaction among these 3 levels of biodiversity. Include in your answer an explanation of each level. All right, so first we're going to explain each one, and then we'll talk about the interactions. So remember, it's 9 marks. You're going to have kind of 9 talking points here. So genetic diversity. Well, this from Unit 1 and CSEC. This is basically like if you have many members of a population and all those members have little small differences in alleles and traits. Right? So differences among members of a species. Dogs, for example, you have Rottweilers, you have Chihuahuas. You have all kind of different potong that have, and all of them, even though there's the same species, they have like a wide gene pool. There's a lot of variation. So because of this genetic diversity, right, there's a lot of genetic differences, a wider gene pool among members. And there's, as a result, now if you have very large populations, you would have even more genetic differences. So larger populations tend to have even a greater range of traits and alleles. So genetic diversity now is the differences between the members of the species. So what is species diversity or species richness? So this is when you have, first to define what a species is. Well, one definition says that, if not counting asexual ones, members of the same species are able to produce fertile offspring with each other. And what species diversity is basically the number of species in a given habitat. So if you have a pond, this would be all the different types of snails, all the different types of fish, all the different types of algae, all the different types of frogs, right? All these different species coming together would contribute to species diversity or richness. This is usually dependent on geographic location, the amount of predators, the amount of food available, the climate, the pH, the temperature, things like that. Ecosystem diversity now is when you have a number of different ecosystems within a geographical area. So, for example, if we say that we have a forested area, that forested area could have a river, it could have a swamp or a marsh next to it, it could have a hilly part, it could have a part with that has acidic pH soil. You have a part that could be waterlogged, you have a part that could be dry. So it also includes larger scale ecosystems like forests, deserts, swamps, but also on a smaller scale within those macro systems. So like within a swamp or a forest, you could have shaded areas, unshaded areas, parts that have water, parts that don't have water, parts that influenced by a water source such as a river and so on, right? So ecosystem diversity now is just the amount of ecosystems in that general area. So how do all of these interact? So the interactions. So populations with high genetic diversity are more likely to survive. So this is because they can overcome selective pressures. If you have, if we take Darwin's finches, for example, they were able to survive on a whole set of different diets. So competition wasn't really a thing that kept them back. And they will say that you have the mutts. Some mutts are black, some mutts are white. And if some of them could camouflage and could avoid predators or avoid being visible to predators, they're more likely to survive. And they're able to contribute to greater species diversity. So high genetic diversity leads to greater species diversity. And higher genetic diversity also improves resistance to disease, pests, reduces risk of extinction, makes them avoid predators, things like that. Ecosystem diversity helps ensure species diversity due to facilitating various factors that help a species overcome selective pressures. So, an example, this would be, let me say that we have a species in a swamp and there's suddenly some kind of predator that appears in that swamp. Well, if there's a forest next to that swamp that the predator cannot go to, the species could flee from the swamp into the forest and escape that predator, and thus it could probably start a whole new family there. But if you have plenty different ecosystems next to each other, you could end up with overlap. You could end up with increased migration between species, and in a sense now that would help them to overcome any selective pressures. If they're feeling pressures in one area, they might not feel one in the next one, especially if they're right adjacent to each other. Then we look at the next one. So question 5 here is 8 marks, so you see I have 8 points. So you read the question. An athlete loses a considerable amount of water by sweating during training, after which he has a high intake of fluids. Explain the mechanism by which water balance is maintained in this athlete's body. So this is a question about the kidneys, the hormone ADH, antidiuretic hormone, and basically osmoregulation. So you look at the points that was made here. First thing you have to note is that there are sensory neurons in the hypothalamus called osmoreceptors that basically detect any changes in the water potential of the blood. If there's an increase, if there's a decrease, and so on. Now, this will enact what is called a negative feedback system. Basically, what it means is that if the water level goes too high, it will try to lower it. If it goes too low, it will try to raise it. Basically, to bring it back to our norm, to bring it back to equilibrium. So, if the blood does not contain enough water, osmoreceptors secretes more ADH into the blood from the posterior pituitary gland. So what ADH does, right, and if we break down the term ADH, right, A is anti, D is diuretic, and H is hormone. Well, we know hormone is a chemical released by a gland that targets an organ or tissue, right? Now, diuretic, you could think of that as a substance that makes you urinate. So an antidiuretic is a substance that suppresses urination. So what ADH does is that it actually suppresses urination, right? It reduces the volume of urine that is formed. So what ADH does is that it takes water from the kidneys and puts it back into the bloodstream. This is in case you don't have enough, you're sweating plenty, it's a hot day, you know. So what ADH does is that it increases the permeability for water in the distal convoluted tubules in the nephron and collecting ducts. So what it does is that it moves aquaporins to the membranes. Aquaporins, if you remember, are transport proteins specific to water. So what this does is that it basically creates a bunch of water tunnels in the collecting duct so that water could basically spill back into the blood. So as a result, more water is reabsorbed into the blood from the collecting ducts. So because there's more water in the blood, the urine would have a lower amount of lower water potential. So the urine is more concentrated with urea and salts and becomes darker and a yellow color. There's like, imagine that you didn't drink enough water for the day. If the blood has too much water, less ADH is secreted or ADH is suppressed. So as a result now, the collecting duct cells become less permeable to water. Right, so there's less aquaporins that move to the membrane. So what happens there now is that imagine that this is the collecting duct. So instead of the water going back into the bloodstream, right, so this doesn't happen. This happens through aquaporins, right? What happens now is that the water just ends up continuing to the bladder, the ureters and the bladder. So this is like if you already have enough water, you don't need to put any back into the bloodstream. You already have enough. So you don't need any ADH here, right? You want to urinate out all this excess water. So it becomes less permeable to water. Urine becomes diluted or clearer because there's more water obviously, and a larger volume is excreted, thus actually reducing the water content in the blood. So this process is known as osmoregulation, and it helps maintain the water potential of the blood due to negative feedback mechanisms. Okay, so B, here we have everybody's least favorite topic, I think, in module 2, which is this action potential stuff, although this one is really just dealing with resting potential. So the question here is, the potential difference across the membrane of a resting neuron is approximately negative 70 millivolts. Explain the role of the membrane in maintaining this resting potential. Alright, so in order to explain this, we have to be able to understand what the membrane of the neuron looks like. Now you're going to see two structures here. So you're going to see this here. So this is actually part of the same, well, this is the same type of structure, and then we have this leak channel here. So these here, these more orange ones, light orange ones, or yellow I guess, these are sodium potassium pumps, and you're gonna see sodium in green and potassium in red. And what's to notice that these are actually positive ions, right? So these are positive, and what's happening here is that they sort of do a trade, and for every 3 sodiums that get removed from inside the cell, down here is inside the cell, for every 3 sodiums that get removed, 2 potassiums move in. So every 3 plus that move out, 2 plus move in. So if you think about it now, this is losing more positives than it gaining. So inside here, you could think of it as there's a net negative inside here then, because this is losing more positives than it's gaining, because every 3 that's moving out, 2 is moving in. So this is what happens at these pumps, right? We need to get into what is happening after and think more concerning with what's happening here in this membrane level. Now there's also these leak channels. These allow the loss of potassium, and only potassium could pass through these leak channels. There's more of these potassium ones than anything. So you get even more positives leaving here. So what happens then? Well, outside here becomes more positive compared to inside here, down here inside the cell. So inside the neuron, there's sort of there's a negative, right, net charge on outside, right? There's a positive net charge outside. Inside here is negative because it's losing all of these. So coming back here now, in resting potential, the inside of the membrane is negative compared to the outside. That's what we just established. The sodium-potassium pump in the membrane removes 3 sodium ions for every 2 potassium ions brought into the cell. So for every 3 that's leaving, 2 enters. This results in a more negative charge within the cell. ATP is used by the sodium-potassium pump to maintain the concentration gradient via active transport. So this actually expends energy, and what it does is that it continuously tries to maintain a concentration gradient, because you could think of it as if it's equal on both sides, you wouldn't get any movement happening, right? The membrane is less permeable to sodium ions and potassium ions. So remember, say you have these leak channels. These potassium leak channels, right, allow the ions to leak out. There's also more potassium gates compared to sodium gates. Once again, this contributes to more movement of potassiums out. And what these contribute to is the movement of more potassiums out, which is of course leading to more negative charge inside here because it's losing positives. Membranes hold negatively charged ions within the cell, thus contributing to the negative charge within the cell. So all of these here contribute to this negative voltage inside. We don't need to go beyond that. All I want to know is how it maintains this resting potential and negative. And it's simply because what happens is that we end up with 3, for every 3 sodium going out, because we're losing 3, we're only getting 2 in return going in. So as a result now, this down here would have a much lower charge than up here inside the cell, right? Would have a much lower charge. All right, so the last question here deals with diseases. Chronic diseases, which are increasing in both incidence and prevalence, are precipitated by lifestyle choices and are having a marked effect on mortality, that's like the death rate in the Caribbean. Explain this phenomenon referring to chronic disease as example. Okay, well, this is 7 marks, and just to define what is a chronic disease, so they are long-term medical conditions. The short-term ones are called acute diseases. And they usually are degenerative, or they may progress into something worse over time if not treated. Examples like type 2 diabetes. They are usually non-communicable, which means that they're not infectious and not caused by pathogens, viruses, bacteria, and so on, and are the result of negative lifestyle choices. This could be like poor nutrition, sedentary lifestyles, lack of regular exercise, but that's a sedentary lifestyle, abuse of drugs, things like that. And so we have, for what is poor nutrition, you try to give some examples: refined sugars, high LDL cholesterol foods, triglycerides, sodium contributes to hypertension, triglycerides contribute to obesity and thus coronary heart disease, atherosclerosis, and things like that. And yeah, so we're just linking it back now to mortality. Well, increased mortality occurs at later stages of life, and more people are being diagnosed with chronic diseases each day, which leads to higher prevalence. And this could be due to plenty of things. It could be due to the advent of technology replacing manual labor, just sedentary lifestyles on the whole. You'll get the idea. And the thing is that these diseases usually help facilitate or exacerbate other diseases. So COVID was a good example. You had all these comorbidities, right? And inflammation of organs, and that actually slowed the immune response to the COVID virus. That's why I saw a lot of people who were obese or hypertensive or diabetic. They had a lower chance of survival against it. So presence of these diseases may reduce immune responses to infectious diseases and opportunistic infections. So that would, of course, lead to increased mortality. And here we have the last question. The last question is 8 marks. Joseph contracts dengue fever and recovers. 3 months later, he goes to another town to take care of his grandmother who is suffering from dengue fever. He decides to take her to the hospital where both his blood and his grandmother's blood are tested. The antibody concentration of Joseph's blood is found to be abnormally high, even though he does not have any symptoms of the disease. So he basically has the pathogen in him, but he's not feeling sick. Discuss why his immune response is heightened. Now, we have to remember here, Joseph already gets this disease. His body already knows this disease. So he has developed, what we will call natural immunity to, not a natural immunity, but a natural immune response to dengue fever. Now, dengue doesn't have a vaccine, so there's no artificial immunity for it, right? So, but basically he got it already. His body is familiar with it, and thus he would be able to fight it off faster before any symptoms could come. So let me look here at different points you want to make to explain this. Joseph is undergoing an immune response to disease. Of course, that's why his antibody concentration is high. So this will be increase in antibodies and leukocytes, or white blood cells in the blood. The immune response occurs following the detection of antigens present in the body, the antigen in this case being on the dengue virus itself. Due to previous infection, Joseph's immune system would have already undergone a primary response. So which means that he would have produced antibodies against the first encounter of the dengue virus. Now, because now he would have produced antibodies, there would have been some plasma cells and some memory cells that would have been produced as well. So his B memory cells specific to that virus would have been formed. What memory cells do is that they retain memory of the antigen. So it's basically saying, I know what that is. Hey, look it coming again. Let me take care before I do anything bad. We already went through this before. So what happened is that Joseph was reinfected with the virus, and possibly from his grandmother. And the memory cells now remember what dengue is, right? And they're able to enact a quick response due to previous exposure to the virus. What this quick response would be a rapid secretion of antibodies from plasma cell clones. They would have been able to multiply very quickly after detecting the virus. They wouldn't wait, they wouldn't try to analyze, but like, we know this already. So this is called the secondary immune response. And this is usually why, you know, when you get a shot, like if you get a COVID shot, you have to get the first dose and then the second dose. Right? So you could build up those memory cells. I could build up this type of fast response. The secondary immune response is faster than the primary response, just as I said, meaning that the viruses will be quickly destroyed before symptoms can manifest, basically before the virus can proliferate or reproduce with anybody. So as a result, now Joseph does not appear ill due to the shortened lag time between detection of the virus, which is basically when his body detects the antigen, and elimination of the virus, when his white blood cells are actually able to eradicate or get rid of any traces of dengue in his body. So that's why his antibody. So if you look at a diagram here, we could see, but this is due to a vaccine, but let me say that this is just exposure to a virus. So if you have exposure to a virus here, you can see that the antibody concentration increases. We get some plasma cells being formed to fight it off, and you have more, of course, because you're trying to get rid of the pathogen. It drops over time because you're getting rid of it, so you don't need as much antibodies. And this would be a secondary immune response right over here. And what's happening here is that you notice that there's a steep increase in antibodies. This is your secondary response. And this is because your body has these memory cells now. And these memory cells are able to recognize the dengue antigen, the dengue virus. And is able to say, hey, we're coming with you with force now. We're going to produce all these antibodies. And as a result now, because we have so much, you could think of the virus getting flushed out your body faster. And you produce more memory cells and you produce more plasma cells, and instead now these would be more long-lived or more long-term. And that's basically what's happening here. And that's it for this paper. Ole take care. Laters.